So polysaccharides, we are saying the first one we should understand, we are talking of starch. And we are saying starch is formed from alpha glucose molecules. We have got one starch which is straight and one which is branched. So when you are forming starch, remember we are using alpha glucose. So let me just show an alpha glucose here. OH is down at carbon number 1, and then we have got OH here at carbon number 4 down. On top we have got hydrogen, and then we have got CH2 OH carbon number 6 there. This is going to interact with another alpha glucose. Everything is still the same. And then I'm going to put just the three of them, that we need to know that it's poly, like they'll continue going like that. So it's a long molecule. So what happens in this molecule? These two are going to interact, forming water. That's uh, a dehydration reaction. Same with these two. They'll continue connecting like that. Remember, this is carbon number 1 of one glucose molecule. This is carbon number 4. So that forms a 1, 4 bond. Because all of these alpha glucose molecules, this is going to be alpha 1, 4, a glycosidic bond. That's how we name them. So I'm going to remove this here to show the bond. Because we have got OH, OH, one OH goes with one oxygen is going to remain. That's the one which is going to lead to the connection of the bonds here. And it will continue going like this. This is why I've just ended like there. It will continue going. Yes, Joyce. Joyce? Yes, go on. Yes. Yes. Mhm. So that forms a water molecule. And in this chemical reaction, water is released. So whenever you are forming a disaccharide or a polysaccharide, one other product is also water. That is why we say these are dehydration reactions or hydrolysis reactions, because they release water. Okay. So the second one, I'm also going to put the same alpha glucose molecules we are seeing. We have got CH2 OH. Now for this one, I'm going to put first this one. I'll show OH there, H, and then also another one like that, H, OH, and then also here I've got OH, H. For this one, I'll put also down CH2O, H, got an oxygen there like that. Then you have got O. For here, I'm going to show an interaction with another molecule that side, because we've already formed the bond just like this one which is here, and it will continue going like that. So this interaction here is also going to form a bond just the same way like that one, no difference. Okay, like that. Now the only difference is, for this other starch molecule, carbon number 1 is also going to interact with carbon number 6 here. So you can see that we have got OH, and then we also have OH there. So those are also going to interact. One oxygen is going to remain. That oxygen which remains is the one which is going to form a bridge between them. So this OH is going to go, also this OH is going to go. We are going to remain with one oxygen. That one oxygen is the one which is going to form a connection between these. So this other one has got branches. So now what's the difference? So the straight chain starch molecule which you are able to see here is called amylose. And the branched one which you are able to see is called amylopectin. So starch has two forms. It has got the linear form that is called amylose, which only has alpha 1,4 bonds. And then it has got the branched one, which is amylopectin, which has got alpha 1,4 bonds and also alpha 1,6 bonds. You can see this is alpha 1,6 because this is carbon number 6 with carbon number 1. But this is alpha 1,4 because we've got carbon number 1 and carbon number 4. So these are the two different forms of starch. Now you need to understand these two. Okay. So if we start first with amylose. Amylose, we are saying, is a straight chain structure consisting of thousands of alpha 1,4 glucose molecules linked together through 1,4 glycosidic bonds. These are thousands and thousands of them. Now, when these are so many, they are going to coil, they are going to form coils. Like when they become too many, you are going to just see them like this. And these are going to coil. You are going to see the shape like that when they become a lot of them. Okay. So when they coil, they are now going to come into, this is what we call alpha helix. This shape you have seen here, these coils you are able to see. And these are now going to go into a compact shape. They are going to become so small to be able to fit in this cell. Now, a suspension, when you get to suspend amylose, this amylose you are able to see. If you suspend it in water, it is going to give you a blue black color with iodine solution. This is why you say that in high school, the test for starch, when you get to put iodine solution, it gives you a blue black color. That is because of the amylose. The reason for that is iodine itself. Iodine solution is a straight molecule like this. So it is going to interact. This form of starch, which is the amylose, they are going to also, like, they are going to interact, they are going to fit into each other. Because of that, the interaction now is going to release this color. Now the color comes in because there is some chemistry which is here. When these two interact and then light is passed on them, light that has got a wavelength that gives you this color is going to be absorbed. Because that light is absorbed, remember light has got different colors. Because it has been absorbed, or the electromagnetic spectrum has been absorbed, that is the color that you are able to see. But for now, don't worry much about that. It's not more our interest. So the total starch, amylose just makes up about 20% to 30%. And I'll repeat, this is biology. No it single statement made. Alright. So, and then we'll go to amylopectin. Amylopectin is similar to the structure of amylose, but it has got branches as you are able to see. Okay, you have got alpha 1,6 glycosidic bonds and also alpha 1,4 glycosidic bonds. When you get to suspend amylose in iodine solution, you are going to see a red violet color. This is basically the color for iodine. And the reason for this is that there is no interaction between amylopectin and and the iodine solution. So in a cell, or just when you get a sample of starch, about 70% to 80% is amylopectin. So what just gives the color of starch is what? Is the amylose. But remember, what is the function of starch from what we've always known? Does anyone remember? What's the function of starch? So starch is used for energy storage. And remember, one characteristic of energy storage molecule is it should be branched so that you can easily break it. That is why the highest percentage of starch is what? Is branched. Because that's the function of starch for energy storage. But the color change is due to the amylose, which is present as a form of starch. Do you have questions there? Do you have questions? So starch mostly is going to be seen in foods such as rice, you talk of maize, wheat, cassava, potatoes. So those are the things. So starch can easily be broken down in a reaction that is called hydrolysis. Remember, when you are forming these bonds, that is called a a reaction in which water is released is called a dehydration reaction. But when you react this compound with water, you are going to break it. And that is called hydrolysis reaction, to breaking a compound with water. That's a hydrolysis reaction. So it can easily be broken. These bonds can easily be broken, forming glucose. And that glucose is going to give you energy. So this is basically on starch. Any question? Okay, since we are good with that. So starch is simply the energy storage form in plants. But the energy storage form in animals is called glycogen. So animals don't have starch, just like plants don't have glycogen. So glycogen is very very similar to amylopectin, meaning that it has got also branches. But the only difference is that the branches in glycogen are a lot. So I'm trying to put down the structure for glycogen, which is just more the same like amylopectin. That's the way we had drawn. So even this side, they'll continue going like that. It's really a long molecule with a lot of branches. Okay, I can end there to continue going a lot and a lot of branches like that. So if we're able to see just the same way amylopectin was looking, that's also how glycogen looks like. The difference is that glycogen has got a lot of branches. So the bonds which are found in glycogen, you have got alpha 1,4 and also alpha 1,6. These are the bonds in glycogen. Now, glycogen exists in form of granules. When you're talking about granules, these are just more like small particles exist in that form. Okay, like that. Another thing to be able to know is that uh, in the cytoplasm of many types of cells, that is where this is going to be found. The glycogen is found, and mostly you are going to find it in muscle cells and in liver cells. And why should it be found in the liver and in muscle cells? Remember, this is used for energy storage, and so you are going to find it in cells which do a lot of work, like the liver. If there is, especially when people get to take in a lot of alcohol, This is going to be a lot because you always need to break down that alcohol. Muscles are always used every time and again in walking, waking, and a lot of things. So that's where you find more of glycogen. Okay. So glycogen is hydrolyzed, or it is broken down into glucose for use in the process of respiration. That is, when you don't have glucose, the option that is going to remain is you need to break down glycogen because it is used for storage of energy. So although it is similar in structure to amylopectin, its branches are shorter and they are more frequent, like they happen most of the times. For amylopectin, you might find that for you to find the branch, it will be after a lot of monosaccharides, but here there are a lot of them. So the glucose chain of glycogen are organized in branches of tree originating from a protein molecule at the center of the structure. So the way we're going to see, I'm not going to remove this since we've already seen the structure. So the way we're going to see glycogen is at the middle, the way it is organized is you've got a protein molecule. You've got a protein like that. And then you have got the glycogens which are originating like that. This is how it is found in cells like that. So this is about glycogen, not different from glucose. It is from starch. It is also a storage molecule. The reason as to why it is branched is because energy storage molecules are supposed to be branched for them to be easily broken down. Cellulose is a structural polysaccharide which is used in structures, and this one is found in the cell membrane. It is insoluble, and I think it's the only polysaccharide that is made up of a beta glucose molecule. The others are made from alpha glucose molecules. Okay, so it is made up of beta glucose molecules, and what you are going to see is, let me draw the structure. You see how it's going to appear. So since this is beta, meaning that the OH is going to be on top, and then the H is down, and then at carbon number 4, you are going to have the OH down because this is glucose. Okay, like that. And then you are going to have also, I'll just put the 3 and then we see how the bonds are formed. Alright, now from what you are able to see, when you are forming cellulose, what is going to happen is, like the way these two are, the bond is going to make more like a Z like this. But because of that, we don't want to form that Z. They are going to be alternating. So what we are going to do is we are going to flip this one upside down. So just the same way it is, we are just going to flip it, meaning that the OH here is going to be on top, the H is going to be down. The OH here is going to be down, the H is going to be on top, and then this O is going to also be down. That is how it's going to happen. So let's flip it. Okay, so it looks like this. And remember now the OH here is down, the H here is on top, and then here the H is down, the OH is on top. And at carbon number 6 here you have got CH2OH, meaning that here the CH2OH is going to be at this point because we have swapped. And then here we've got CH2OH. So what you are going to see now, these are going to interact, these are also going to interact to form those bonds. Yes, please. So you just need to know if they give you such a structure. That's how it looks. So Because biology is all multiple choice, so you'll not be allowed to draw. If they give you such, just know that it's the one. How do you tell that it is alpha? I mean beta. Look at this oxygen. Whenever hydrogen is down this oxygen, just know that what you're talking about is what is beta. But if the hydrogen was on top near this oxygen, then that's alpha. So here we have got beta 1 comma 4 glycosidic bonds. These we are able to see here. Beta 1 comma 4 glycosidic bonds. And for cellulose, the H2OH, they're always alternating. In one, you find it on top, find it down, you find it on top, down, like that. Okay. So the other thing is that these are unbranched, they don't have branches. So cellulose doesn't have branches. Remember, it is used in structures, it's used in the cell wall. The OH group, which project outwards from each other, form hydrogen bonds. These, at this point, we have got an OH. Okay, these OHs you are able to see. You are going to have, so in a simple way, I'm going to remove this in a simple way because these are very small, like when you are viewing with a microscope. You're just going to see lines like this. Then this is one cellulose molecule. You are going to have another cellulose molecule. We had OHs at the ends of these. So those OHs are going to form hydrogen bonds. They're going to attract another cellulose molecule like that. When these get to attract each other, they are now going to more form these. When they form a lot of these, a lot of them, that is what now leads to the solid structures which are able to form what? Form protection layers in structures. So we are saying these OHs, they are going, which project outside, they are going to be attracting each other via hydrogen bonds. Neighboring cellulose molecules lie close together in layers and form rigid structures which are called microfibrils. So these which are able to see here, when they come together like this, these are going to form what we call microfibrils. And when these microfibrils, they come to be arranged together in bundles, like you have got another one like this, and a lot of them like that. So together these two microfibrils, they are now going to form what is called macrofibril. So these bundles are arranged in layers and they have tremendous tensile strength. Like they've got such greater tensile strength that you cannot break them. You can just maybe fold them. So the cellulose layers are fully permeable to water. They allow water to pass through. That is why we say the cell wall is fully permeable because of what? Because of the cellulose layers which are there. Cellulose is the most abundant organic compound on Earth and is a major structural material of plant cell walls. That is again what we should be able to know. So this is cellulose. Now what you are going to be able to see is we have talked of microfibrils and fibrils. So you can have this structure like this. So inside you have got small ones like that. So these you are able to see inside here are what we call microfibrils when the cellulose molecules are together. And then those microfibrils, when they come together, a lot of them, they are going to form a fibril, which is a macrofibril. So this is how they are. So you can talk of, when you're talking about cattle, horses, and just other ruminant animals, as well as just termites, they have got bacteria within them, and those bacteria within them, they are able to break down this cellulose. Like cellulose cannot be, like in humans, when you get to eat these, you don't have bacteria which can break them down, but for cattle and horses, they've got those. So they are able to digest these things. Okay, when the bacteria which is found in the cattle digest these, that is what we call symbiosis, a relationship where the bacteria is benefiting because it is finding an environment for them to stay. That is the environment in the what? In the cow. And then also the cow is benefiting from the what? The cow is benefiting from the bacteria. Okay. by when the bacteria gets to break down these molecules. Sorry, I've run out. My battery percentage is low, so I'll forward you the video at the end. Okay. So when you're talking about cellulose, cellulose is important in human diet as a dietary fiber. It's used as a dietary fiber. That part of vegetable that you cannot digest, that's a dietary fiber. That's the importance of cellulose. And cellulose is used in the industry to produce paper and also cotton. All right. So the last polysaccharide that you should be able to understand is chitin, which is found now in animal exoskeletons. Now, chitin is more like cellulose. The only difference is when you look at cellulose, the structure, of course, we drew it already. Okay, so this is how cellulose was. That's how we drew it. And then we had alternating bonds. This was CH2OH. Even here we are going to have CH2OH. Okay, oh, this one I've also drawn it wrongly, so the oxygen is supposed to be at this point, and then we're going to have CH2OH. At carbon number 2 of glucose, we're supposed to have an OH. But this OH which is here is replaced by a carbamide group. I mean acetamide. The acetamide, you just have NHCO CH3. This is a group that replaces carbon number 2. Even at this point, the same group like that. Even at this point, the same group. So this is the only difference between chitin and cellulose. Just carbon number 2. Instead of having OH, you have got acetamide functional group. So chitin is an important unbranched polymer, and it is found in the cell of fungi, exoskeletons of insects, crabs, and shrimps. It is similar to cellulose, but the hydroxyl group of carbon 2 of each glucose has been replaced with an acetamide. So that's the only difference between cellulose and chitin.